For most of the 20th century, developmental biology operated under a strict rule. Cellular differentiation was a one-way street. Once an embryonic cell matured into a specialized skin, muscle, or nerve cell, its fate was locked in a terminal state. This rule created a scientific and ethical bottleneck. To utilize pluripotent cells, which are capable of becoming any tissue type, researchers were forced to harvest them from human embryos, triggering intense regulatory friction. In 2006, that assumption collapsed. Researchers discovered that specialized cells could be forced backward. Their biological clocks could be rewound to a primitive, embryonic-like state. The breakthrough came from Dr. Shinya Yamanaka and his team, who proved that a cell's identity is entirely fluid. By isolating the specific signals that maintain pluripotency, Yamanaka's team bypassed the need for embryonic stem cells. They demonstrated that the chemical markers dictating a cell's age and function could be actively rewritten. The path to that discovery began in 2004 at Kyoto University, under an initiative internally known as the Bingo Project. The researchers started with 24 candidate genes known to be active in embryonic stem cells. Using retroviruses, they shuttled these genes into adult mouse fibroblasts to see if any combination could trigger a reversion to pluripotency. To test the results, they engineered a selection mechanism linking the FBX15 promoter to an antibiotic resistance gene. During an antibiotic wash, specialized cells died. Only those resetting to an embryonic state activated the shield, survived, and formed colonies. Through a systematic process of elimination, removing one gene at a time, narrowing the list from 24 down to 4 essential factors. Identifying this 4-gene code proved that cellular identity functions as programmable software running on the epigenome. This table outlines those 4 factors, abbreviated as OSKM. They are transcription factors, meaning they bind directly to specific DNA sequences to control the rate at which genetic information is transcribed. OCT34 is the indispensable core of the group. It acts as the master regulator of the pluripotency network, locking the cell in an unspecialized state and preventing it from developing into early embryonic tissues. SOX2 acts as the stabilizer. It partners directly with OCT34 to create a continuous positive feedback loop, maintaining the cell's ability to self-renew without differentiating into neural tissue. KLF4 facilitates the physical transition. Genetic reprogramming puts immense stress on a cell, which normally triggers a defense mechanism called the p53 pathway to halt the cell cycle. KLF4 suppresses this pathway, keeping the cell alive through the process. Finally, c-MYC acts as the catalyst. It physically opens up the dense structure of the DNA, a process called chromatin remodeling, so the other 3 factors can easily access their target genes. Together, these 4 proteins form an interlocking regulatory circuit, powerful enough to forcibly overwrite a mature cell's existing identity. But translating this biological mechanism into medicine presents a paradox. c-MYC makes reprogramming efficient, but it is a known proto-oncogene. Overexpressing it drastically increases the risk of tumor formation. Furthermore, complete in vivo reprogramming is catastrophic. If a specialized heart cell fully resets to a stem cell while still inside a living body, it wipes its epigenetic memory. The tissue loses its functional identity and fails. To solve this, researchers targeted the actual mechanics of aging. Over a lifespan, cells accumulate epigenetic noise, disorganized chemical markers that suppress cellular function and drive decay. Instead of fully reverting the cell to an embryonic state, scientists developed partial reprogramming. This involves a highly controlled, transient expression of the factors. By utilizing an OSK cocktail, omitting the dangerous c-MYC entirely, a temporary dose safely clears the superficial epigenetic noise. The cell maintains its specialized identity while functioning like a much younger cell. Partial reprogramming redefines aging as a reversible epigenetic condition rather than inevitable physical decay. The shift from theory to living tissue was validated in 2020 at Harvard Medical School's Sinclair lab, targeting age-related vision loss in mice. Researchers packaged the 3-factor OSK cocktail into an adeno-associated virus, safely delivering these instructions directly into the nucleus of damaged retinal cells. The resulting data showed a safe 5-fold increase in nerve regrowth, the mice experienced a measurable reversal of vision deficits without any signs of tumor formation. Other firms are developing non-viral methods. Stanford University spin-out Turn Biotechnologies approaches the problem using a targeted protein bath. By administering precise, transient exposures of the Yamanaka factors to aged cartilage from osteoarthritis patients, the cells ceased producing inflammatory markers. Similar results occurred in impaired muscle stem cells, restoring their regenerative capacity. These clinical models confirm that the molecular architecture functions in complex living systems. The biological age of differentiated tissues can be safely reversed without compromising structural integrity. As the clinical data matures, the intellectual property controlling this technology is shifting in real time. Since 2006, Kyoto University's IPS Academia Japan has guarded the foundational OSKM patents. Those original patents carry a 20-year term, meaning they expire in 2026. This breakdown of their 2025 portfolio shows a clear pivot. Patents for basic reprogramming techniques are shrinking. Instead, massive growth is occurring in specialized differentiation processes and direct therapeutic applications. This patent expiration is driving heavily capitalized startups, such as Altos Labs and New Limit, to bridge the gap between academic theory and large-scale therapeutics. New Limit uses machine learning and single-cell genomics to map out entirely new transcription factor combinations. By finding alternative proteins, they aim to achieve epigenetic rejuvenation while bypassing legacy patents and oncogenic risks. The biotech sector is no longer focused on simply learning how to create a stem cell. The capital is now deployed toward the industrialization of targeted cellular rejuvenation. The final challenge lies with global regulatory bodies, which must adapt traditional drug approval pathways to govern living, epigenetically altered cells. Japan maintains a proactive framework. Their 2014 PMD Act established a time-limited conditional approval system, recently resulting in the commercial clearance of Amchepry, a reprogrammed cell therapy for Parkinson's disease. This matrix shows Japan balances speed with post-market accountability, allowing early revocations if data fails. Meanwhile, the U.S. moves conservatively. Under the FDA's RMAT designation, treatments clear high evidentiary bars, recently moving the reprogrammed therapy Fertilo into Phase 3 trials. The FDA requires exhaustive safety screening of off-the-shelf allogeneic cells to prevent genome instability, ensuring patients do not develop clinical side effects like severe dyskinesia from poorly integrated tissue. Meanwhile, China deployed regulatory reforms in 2025 to shrink clinical trial approval timelines down to just 30 days, rapidly expanding domestic commercialization pathways. The Yamanaka factors resolved the ethical controversy surrounding embryonic research, while providing a mechanism to control biological time. This insight initiated a global race to convert the reversal of aging into a standard medical protocol.